back a bit ourselves. All right, brilliant. I think that's good enough for now. Okay. Good afternoon, everybody. Wow, the light is very bright. Good afternoon, everybody. Thank you for joining us either online or in person. We've been waiting for this talk for about a year now. This is Phil Holland, who's one of our lecturers here at King's, and he also works with a number of organisations, and his research is all in migraine and pain. And he's here to give us a very interesting talk today about uncovering brain circuits in migraine. So Phil, take it away.
Thank you. Thank you for the invite. Sorry it took so long. I've been busy. So I'm on the board of the European Headache Federation, and shortly actually going to become the president of the European Headache Federation. So I've been keeping myself busy, but based here at the Wilson Spark on the Guy's campus. And we're going to talk a little bit to start just about migraine to give you the flavour of why we're interested in migraine, and then change that into why I think neurotechnology is really important for modern neuroscience, especially in terms of what we do. So headache itself, I mean, I'm sure most of you will have suffered from a headache to some degree, about 90% of the population will. Migraine, 1 in 5 people, 3 times more prevalent in women than men. So considering that neuroscience is now dominated by females, certainly King's undergraduate neuroscience has a high prevalence of migraine there, or expected. Chronic migraine, this is not like chronic pain where the pain is constant. It's where you get migraine attacks on headache attacks on more than 15 days per month, 8 of which are full-blown migraines. So it's about the frequency of attacks. Again, can affect children, and really about 1 billion people globally. 10 million in the UK suffering from migraine diagnosed, and that's a big number, but it's put better this way. That's equivalent to the population of the USA and Europe combined. So if you combine the population of these two large areas, then that's the number of people who are diagnosed with migraine, just slightly over that. And many of you have heard about migraine in the past, might have known migraine has been proposed to be a vascular disorder. That is, dilation of blood vessels and cause the throbbing pain, and we know that's not true anymore now. We know that because during spontaneous attacks, this is a human here, and you're looking at vasodilation, and you can see really no dilation in the vessels during attacks. And very simple study, not a neurotechnology study, a very simple study. If you measure a patient's cardiac output, so their pulse, and you also get them to tap the throb rate, when the pain's throbbing, you can see the heart rate is the red, the throb rate is the blue. They occur at completely different frequencies, actually. So the throb rate is completely independent of pulse rate. So we've become very interested now in how it's actually neural circuits that regulate that, normal brain cycles that are regulating that throb rate, not vessels. And really brings us to this point that migraine is now a disorder of the nervous system. And that'll give you an idea of where neurotechnology comes in, because we need these neurotechnology approaches now to be able to dissect the nervous system in conscious living organisms that have obviously an intact peripheral and central nervous system, can feel pain, and can obviously have all the different cell types that we're interested in. This is the complexity of migraine. So this is now migraine, the individual attack. From over on the very far left, patients, even before the headache, which is the red line, get these very early premonitory symptoms. Fatigue is a very common one we'll talk about today. Photophobia, sensitivity to light, touch, different things. In about 30% of cases, the yellow line, you'll get this aura. That's the flashing lights that some people so often call that classic migraine, but it's really only present in about 30% of people. Can be motor, can be sensory. Then we get this building headache, all the way up to a severe unilateral throbbing pain. And then during the resolution phase out here again, even after the headache's gone, the patients will feel washed out for up to a week, where they just simply can't function. So really this whole attack is probably about a week and a half long in terms of what it's doing to the patients. And this is where we come in now. The diversity of these symptoms suggests that there are a number of pathways involved in this. Patients will report obviously the pain, but also brain fog, cognitive challenges where they can't concentrate, arousal, appetite disruption. So we're really interested now in identifying which circuits underlie these symptoms. If we look at what we know from human imaging, well, during these early premonitory phases, we're really focusing on areas like the hypothalamus. this homeostat for the brain. It's the thing that, links your sleep-wake patterns with night-day. It's the thing that links your energy consumption with your energy expenditure in terms of controlling your appetite and feeding. And we think this goes awry in migraine and leading to this disrupted response. In terms of the aura, we know that to be a very cortical event. It's this spreading wave of depolarization that happens over the cortex, causing these visual flashes when it happens in the visual cortex, as you see here. And then when we go into the headache, we start to recruit the classic pain pathways like the thalamus, and these pain pathways going through up to the cortex, and for that sensory and emotive component of pain that you'll have learned about in your different lectures. And then interestingly, during the postdrome, we see a very different picture now, where because of the high energy demands that's happened in the brain during the early phases, what we see now in blue is a global reduction in activity within the brain, where it's really probably a recovery phase, where it's recovering from that really high energy demand, reduced blood flow, reduced activity, driving a lot of these feelings where the patient feels, again, a bit of brain fog, but also cognitively and energy deficit in those patients. What we mean by premonitory symptoms is this here, is that actually, if you ask patients how they're feeling, this is an electronic diary study and they have to put it in, it's time locked, and the patients are feeling absolutely fine. You see that there, the green line. Then about 24, 48 hours before the headache, which is the red dotted line, they start to feel things are not quite right. They report these feelings. It gets worse during the headache and then improves after the headache resolution. And if you ask these patients, what are you feeling? The majority of them will say fatigue, but about half will say photophobia, concentration difficulties, stiff neck, really common actually. That's why a lot of people go to the chiropractor and get twisted and pith, but it's not really needed. It's a symptom of the disorder. And phonophobia. So you can imagine, obviously, if you have a migraine attack in a lecture, you can't be around, you have difficulty concentrating, you can't have bright light, you can't have loud sounds, you're feeling uncomfortable, you're tired. It really affects what you can do in life. But what's really nice from a scientific perspective, if you ask those same patients, can you predict your next attack based on the presence of these symptoms? Then again, pretty much the same results. About 3/4 of patients can predict accurately the ensuing attack, telling you that these are early disease hallmarks. So these are actually underlying biology of this disease starting long before the pain. And therefore our idea is, well, rather than trying to mask the pain with analgesics, which can often be addictive component, can we understand the circuits that give rise to these features and stop the attack in its tracks before it gets to the headache? So actually treating the disorder rather than masking the symptoms. This is just to work on the hypothalamus and during the premonitory phase. And what was really interesting about this work, for example, was patients who had occipital cortex activity more likely reported light sensitivity. Patients who had periaqueductal gray activity more likely reported nausea as an associated symptom. And again, the hypothalamus, which I'm going to talk about now, polyuria, mood changes, appetite changes, all these homeostatic functions that the brain works at. And this is the interesting bit actually from our perspective, because most of these premonitory symptoms here, now called prodromal symptoms, because that's what they're called in the US for this review paper, are actually related to what you would report as common triggers. So you'll often say, oh, when I exercise, I get a migraine, or when I eat chocolate, I get a migraine. And actually the truth is no. The truth is that chocolate is a craving. And therefore you go eat the chocolate, you're getting the migraine attack anyway, and you link the two together. But the truth is you're going to have the migraine anyway, so enjoy the chocolate. The attack's coming nonetheless. Again, things like, people with stress will report mood changes as in the prodromal symptoms, so it might be a feature of that. People who have sleep deprivation as a trigger might report sleep disruption during their premonitory symptoms. So we're trying to really dissect out what is a premonitory symptom and what's a real trigger of migraine, so that we can understand that also. This is the complexity now, is that having a whole brain disorder means we need to be able to study the brain. It would be lovely if it was just a simple peripheral thing, because we could study that, but it's not. It's a brain network disorder, and therefore we need to study brain networks. And this is where neurotechnology comes in. where we try to look now at the brain as a whole and understand what's giving rise to all these different features that come with your migraine attacks, from the aura, which, remember, are these flashing lights, to the allodynia, the sensitivity to touch, the headache itself, and the sensitivity to light, sound, smell, and we'll talk a little bit more today about fatigue, because we think that's a real driving feature of these attacks. Right, now to the more exciting bit for you guys. Well, how do we do this? Well, I like to explain this simply. Think of your nervous system as a conversation. If you're talking to someone, like two cells are talking to each other. But we can do that. We can record from this, so we can put in a single electrode like we used to do, and we can record this conversation, and we can see how one cell, for example, is responding to another cell. But in doing so, we miss a lot of the conversation, because that conversation is very rarely one-to-one. It's actually a circuit where a number of people are talking at one time. And clinically or pre-clinically, we can measure that conversation with EEG. But again, what we see is the general noise and the general conversation, but we lack the detail. So we don't have the detail of that conversation. We get an idea maybe whether it's a happy conversation or people are moaning or but we don't know the context of that conversation and how the individual cells are responding. So what we've been working on a lot now is using open source technologies, much more affordable, a lot more coding, but more affordable. So an open source electrophysiology rig would cost you about maybe £12,000 to £15,000. The same bought off the shelf system would be well over £100,000. So it has an affordability as well in terms of reproducibility and people developing their own careers. So we can do that with circuits. What we're going to talk about a little bit more today really is this second component, where now we can take these conversations and we can label, in this case using fluorescent dyes or calcium sensor dyes, specific populations here in green of that conversation. And now we can use different imaging technologies to image those cells and look at how they're responding within the circuit and what happens when we change that circuit. And what we've been doing recently now is basically combining both those approaches. We've been looking at this case, this is an example of some of the thalamic work we did, where we were using open eFizz, so 126 electrode channels to record all the neurons in that region, and at the same time recording from these neurons, which are labelled with an activated dye, so that we can look at that conversation from two parts. So can we target now therapies? What we're doing with these tools, so I'll show you this. This is orexin. And if you've done lectures on sleep, you'll know about orexin. It's the thing that controls the flip-flop switch. So it's the on button for keeping you awake. It promotes arousal without keeping you awake itself, so it controls noradrenergic arousal. And if you don't have orexin, if you lose these orexin cells, which are made exclusively in the hypothalamus, you have narcolepsy. So they get destabilization of that switch. So we've become very interested in this pathway for two reasons. So in the rodent, again, it's selectively expressed within the hypothalamus, so it's easy to target relatively. And it expresses then its axons and sends signals throughout the entire brain, where it controls many functions, from nociception as well, but respiration, stress and anxiety, really important for migraine, feeding, wakefulness, arousal, and motivation and reward. So we've been very interested in this pathway and how it can match into the features that we see in migraine. And not the focus of this talk, but if we use chemogenetics, so we use, for example, viruses with a chemogenetic inhibitor that allow us to turn off these cells. If we do that, and this is using allodynia, so in this case we use a little filament and we poke the mouse on the forehead, and the mouse is free to move away from that filament when you want. If you give that mouse something that makes it, say, sensitizes the mouse will move away at a lower threshold. So basically it's responding to lighter touch. It's been noxious, so it's allodynic, just like migraine patients would report during their attacks. And black would be a sham vehicle, and red would be inhibitory dreads. So if you give an inhibitory dreads vehicle to these animals and turn off those orexin cells, you can make those animals much more responsive, so they become much more sensitive to touch. And if you give those animals orexin A in the blue box, you can recover that phenotype. So you can actually see that you can use this to dial up and down the pain response. But importantly for our work, if you just look at the mouse behavior, in this case in an open arena, and you video the mouse, and you look at its activity as a sort of a readout of fatigue or motivation, again, you can see that when you turn off these orexin cells, the mice become less motivated, move less, and you can recover that by giving orexin A. So we've got a hint that this might be an important molecule in terms of this fatigue that we're seeing in migraine. So what do we do next? Well, we look at cells, and you can look at cells down a microscope and tissue, and many of you might do something like this for your undergraduate project. Or you can go to the macro scale, which is brain imaging, so 3 Tesla, 7 Tesla, human or animal imaging. And that's great because we're all very good at them, and the university has great facilities for that. But here's an example. This is some work I did in Edinburgh, actually, over here on the far left. As you look at it, that's looking at nodes of rhombe, so you can image those cells. Or again, you can look at rodent brain. This is a rat brain here again, and you can see and look at different activity patterns. So very strong, lots of people do it, but you can see the old problem again. This is too focused, and this is too global. We don't get that conversation in the middle. This is where our different imaging tools come in. For those of you that are here, I've sent round some samples, so you've seen them, of the little cameras we use, so you understand what they look like. And for those of you online, you'll just have to find out. But there's 2 options we can use here. So for this mesoscale, so we use fiber photometry. Again, an open source technology that's been developed that we've adapted in the lab. And in this case, you label the cells. If I have a point here. In this case, you label the cells in the same way, but now instead of recording from all the cell, one cell, you're recording from the whole population of cells. So it's looking at population level activity. It's kind of like a gross way to look at local circuits. Or with those miniscopes and the grin lenses, and this is what I sent round earlier on, this would be that little glass lens that you were looking at in the tube. You can now image these cells and actually see them individually and record them outside the brain, because that fibre optic cannula will send that information to a camera that attaches to the head, and then you can image that in the live behaving rodent. So you can start to understand how these circuits respond in normal behaviour and in disease. Here's some examples of what they look at. The one on the left is a commercial system in Scopics. That's about £140,000 to buy, and it's about £20,000 per year in license fees in terms of the software to run it and maintenance and stuff. The one on the right is 3D printable open source miniscopes. That's what I sent round. These are the ones from UCLA. So these are, you can 3D print these and build them yourself entirely out of all the components. And you know, each camera then in the system to do that costs probably no more than 20,000 in total to do that. So it allows you this flexibility. Say if you were a young neuroscientist wanting to go start your new laboratory, instead of having to get funding for a couple of million to build the equipment, you can now do it for much more affordable. And because it's more affordable, more laboratories can do it, so therefore we can work on reproducibility. We can use the same cameras in the same laboratories instead of some black box that's got a mystical code in it that we don't quite know how it gives us this number at the end. We have to program it and share those codes via things like GitHub or different pathways. So it's a very shared, it's kind of an open source technology. Things to consider. Well, if you're going to do this, you need to then think, well, how do I label my cells of interest? What calcium indicators? You remember, of course, that cells use calcium when they're becoming active. So we often use these calcium indicators. We're up to about GCAMP 9 now, I think, in terms of definitions. How are we going to get that in there? Do we use breeding? Do we use viral approaches, lentiviruses? And can we get transgenic mice? So in our most recent work, we are crossing orexin Cree mice with GCAMP 6 flox mice. So now we don't have to do injections. We've just, the mice constitutively express the calcium indicator in orexin neurons. So it means we don't have to do that extra stage of injections. You've got to, if you're doing the injections, think about your location, how much virus you're going to put in, and make sure you're in the right position, make sure you're labeling the right cells. Of course, that takes a bit of time to work out. And then you've got to think about the surgery. You guys here saw that little tiny lens. Well, can you imagine that's it there implanted into the brain? It's really quite delicate surgery. And when you're putting these lenses deep, up to, say, for the hypothalamus, 6 millimeters into the brain, it's a reasonable depth. So you've got to then think about head fixed or free moving recording. So for head fixed, we have our animals fixed with a little bar, and they're on little treadmills, 3D printed treadmills, so they can still run and things, but they're head fixed for stability. Or we have them in a cage where there's a rotor arm, and they can run around, and the cable flexes so that we can record the signal. And then analysis, how do we develop that package? How do we make up for motion correction? Because if the animal's running around or you get changes in cerebral blood flow, that affects the motion of the brain and the activity you see. So these are the two approaches we commonly use in the lab. In terms of resolution, the grin lens UCLA miniscope is by far the best because you can see the individual cells compared to the fibre photometry. The fibre photometry is much more sensitive because you get a population level circuit, so that gives you advantages there. In terms of weight, the fibre photometry is much lighter because it's just a small cannula. It doesn't have that big camera attached to the head. I say big camera, it weighs like 2.2 grams or something. And in terms of scalability, the pi photometry, the fibre photometry is much more sort of readily adoptable within laboratories. The grin lenses take quite a bit of development in that way. So these are things you would be thinking about in terms of if you wanted to build a multi-region collaboration or record from different regions of the brain. Pi photometry is much easier. These grin lenses would only really allow you to record from one sort of nuclei at a time and one target cell population. So if you were in a lab and you were thinking about this technology, these are the decisions you would have to make. This is kind of what we're going to be doing in the lab. This is what it looks like on paper. You see this lovely picture in the journal and it looks all great. This is what it looks like in real life. This is how we wire it up in the lab. So this is a breadboard with all the different fibre optic components, light lenses. So this here, this is version 1, so we've got the newer version 2 now actually, but this kind of drives the LEDs that cause the fluorescence and records that signal. This is the pi photometry board that works in with OpenEFizz, and you've got to be able to do all this wiring and soldering and all this kind of work. So although you might see it in a paper looking like this, in reality this is what it actually looks like in the lab. And then you can take that, you implant those probes, and now in this case, for example, you can see these are orexin neurons here in purple, and you can see the ones that are transfected in this one via injections with GCAM6. So you can see you can start to record. There's your cannula down there through the brain, and you can record this population down here in green of neurons. When you do that, what things can you see? Well, this is an example from a paper I took because I think this is a really nice example of why this is also important. Is if you do mechanical stimulation, so this is a head fixed animal and doing mechanical stimulation, in this case a pinch of the tail, what you can see here that's really important is that these cells only respond in the awake animal. Anesthesia has such an effect on the brain's physiology, and a lot of what we've done classically has been done in anaesthetized animals. So a lot of what we've done historically doesn't really tell us the true picture actually. And that's where modern neurotechnology is really useful. Because now, for example, if you do this in an anaesthetized animal, you see no response to that level of mechanical stimulation. Whereas if you do that same thing in an awake animal, you start to see now this population response. So we need to think about doing this in conscious awake animals so we can understand these brain circuits. So now when we want to go to the single cell level, that's the little camera I showed you earlier on, you saw the little examples of them, we can plant those grin lenses. And that, on the other hand, now, very similar thing, we implant a probe, but now instead of looking at the population level, what we can start to do now is we can start to record from individual cells in that network. So here there's 5 different cells labelled and different colour coded, and you can see an example trace from each cell. So we can look at how each cell is responding now within that network, and instead of looking at the global population. So if you want to understand the entries of a circuit, this is a really good approach. And then this is just some lovely work that came out, I think it was last year or the year before, that came out using this tool as in migraine. So I thought I would use this as a nice example of what we did. So this was with the grin lenses and the cameras, so we implanted these down into the trigeminal ganglion. So put these lenses. So you can see this is obviously a very long lens because it's gone right through down into the trigeminal ganglion, which sits underneath your brain, in rodents anyway. And from that, you can then record these, there's baseline. If you pinch the animal, you can see activation of cells, and you see that response. And if I show you it now, do I have a picture on that one? No, there's not a video on that one. On this one now, if we take that same animal and now we put capsaicin into the system, so we inject capsaicin as an irritant that would activate the cells, you can see that here. Capsaicin activates these cells that you're recording through the ganglion. And you can see that down here in real time now with the camera system. So, see them flashing away here. So these are individual cells now in the trigeminal ganglion responding to that sensitized activity that you can see. So you can start to see how these individual neurons now are actually responding to that stimulus response after the capsaicin infusion. And you can, in the same animal, you can look at a baseline response, and this is using a GCAP6 fast, so just an earlier form of the GCAP. So you can see how now we can start to individually record these single neurons. What they were interested in doing is this phenomenon of spreading depolarization, this migraine aura that I spoke about. It's this wave again of hyperpolarization across the cortex, and it causes these visual phenomena, and then you get these areas of inactivity because of the excitability. And often it's visual in the people that report this, and people can also report this kind of prolonged inhibition. So people can get sort of paresthesias in the limbs, or in the case of some people, they get muscle weakness, so they'll get actual motor weakness down one side of the body in response to these if they're in the motor complex. So what they did here now was they put a tracer, so in this case they put a fluorescent tracer into the cortex, and they wanted to ask the question, well, what happens if you then induce cortical spreading depression in terms of the solutes in the brain? What happens here? And what you can see here now, get this video to replay, you can see here they're equal at the same time. This is control with no cortical spreading depression. This is the animal that's had the cortical spreading depression induced. And you can see that that spreading depression causes clearance of the solute much quicker. So it forces a lot of the CSF and solutes from the brain to exit the brain much quicker, which is important, probably by swelling and different mechanisms. And why that's important is because actually, if you then look in the composition of those molecules, so they were interested in what the composition of this was, so they looked at what proteins and different inflammatory mediators were in those CSF proteins. And the important one for us here is this calca-8. That's for CGRP, calcitonin-G-related peptide, this important target for migraine. If you then take that CSF here and you take the CSD, so you take the animal, you induce the CSD, you take the CSD from that induced animal, and now you put that on a naive animal that's not had CSD. So this is it here, you're taking that CSD, you're putting it on the trigeminal ganglion of a naive animal. What you see now is that the animal, the yellow now here is the animal that's had the CSF from the other CSD animal. What you can see is that you're able to passively transfer that sensitization of the system from one mouse to another. And the work went on then to show that this was driven by CGRP. So when you have that event, you get this release of neuropeptides that then when you put them, they get cleared from the brain, they exit onto the trigeminal ganglion, this peripheral pain nerve, and they can sensitize that pain nerve, driving activation of the pathway. So it's only by using these sort of camera systems that we can able to video and understand these mechanisms and understand what's going on in these circuits. You can see that here, that's the response. So the number of cells responding to the CSD versus the naive animals of their own with just naive CSF. So you get this passive transfer of sensitization. And what this work was really able to show was an entirely new pathway that people hadn't appreciated before in migraine now, where events going on in the central nervous system here could, via efflux of CSF and different solutes, affect the peripheral nervous system. So in this case, was able to sensitize the peripheral nervous system and cause activation of those incoming pain pathways. So showing a direct link between the migraine aura that people experience and the potential to activate the pain pathways, how that might be explaining why migraine aura in a subpopulation of people can drive the pain of headache and migraine. And really, it's allowed us to look in much more detail now, because we can image deep into this tissue, and we can see what's happening deep in this tissue and what's happening with these different cell components. So with these new technologies, we've been able to understand much more about these neurovascular units and the cell competition in the brain circuits, both in terms of pain processing, but also, as I showed you earlier, in terms of understanding how these, say, the orexin circuits, what happens to them if you stress, for example, what happens to them in sleep disruption, and what happens to those in people with migraine in terms of how they respond, and why is it that they have this dysfunctional response where they respond. Essentially, think of the hypothalamus as predictive coding. It gets used to your regular routines when you eat, when you sleep. I mean, many of you guys have regular routines. I don't know how, but we creatures of habit. We tend to eat roughly about the same time of day, depending on where you come from. We tend to sleep roughly about the same number of hours. The migraine brain, what it doesn't like is change. It doesn't like to change any of those situations. So we're starting to use these tools now to understand how, through the hypothalamus, when you expose your body to these changes, the hypothalamus does a mistake in predictive coding. And now, for example, instead of encoding the light that's above your head as being, well, it's okay, it's a bit spotlighty, it's not comfortable, it's now misencoding that light as being really stronger than it is. And therefore, you find that light aversive, you find that light painful on top of the migraine pain. I think that's probably my last slide, no? That's just showing the pathway out from the trigeminal ganglion to the dura mater. And this is actually an old slide, so I need to update it because only one person on this slide is still with me. It's Eloisa. Alejandro's gone to Manchester. And, oh no, two. Veronica's still with us. Veronica's a PhD student in the lab. Hui's gone into industry, so she worked with us doing some of the phys that you saw there, but she's gone into industry. Taylor's moved to Birmingham. She works in more cellular stuff, and Ellie's just finished her MSci in Glasgow actually, and was potentially coming to do a PhD with us, but it isn't at the moment. So I have a pot of money looking for a PhD student at the moment to come and do some of this very stuff in cluster headache and associated headache condition. So I have a pot of money for a home student to come in and do a 4-year PhD in the laboratory. Should have been starting last October, but now we'll start next student intake probably June or October 26.
Is it the same opportunity advertised at the end of the migraine lectures?
No, that's the MRCDTP. So that's they're like a program, and we've got money through a program. This is independent funding. So the money for these PhD here comes from a donor. Actually, it's a family who have made a private donation to support cluster headache work, and for family reasons. So we got a few pounds from them to do some work. So we've recruited a postdoc and 2 PhD students, one of whom is going to Leiden. She's already recruited. She's going to Leiden to work in a human post-mortem study. So she's going to be looking at migraine control and cluster headache brains and trying to understand what's changed in those networks in the human brain and how we can then take that back to the animal studies to understand how those circuits work. So trying to link the human to animal work in sort of a translational sense. So that was a whistle-stop tour of how we've started to use neurotechnology within the lab. As we get the different ones set up, the Grin lenses are kind of set up, but probably early next year. Individually, for certainly for people here, if you just want to come and see them or that, reach out to us and we can let you come and see how they are in the lab. You know, it just depends of matching you up with, say, the PhD student that's doing an experiment that day, and you can then come and see how the things work. I'm very happy to do that. Or maybe a little group of 2 or 3 of you to come together and just see the setups and stuff. They're just on the Hodgkin building at the back of Guy's campus.
Very familiar.
I had a quick question about a couple of things came to mind, but with what we know about migraine, and like if you have oral with your migraines, or if you have, I don't know, physical symptoms in your limbs and bits and pieces, we're talking about it being wave of hyperpolarization across the brain, and that's what we think is the cause for that. Do we know why those manifest as visual or sort of proprioceptive symptoms? Is that because it originates in like the cerebellum or the occipital lobe?
No, exactly. It depends on where in the cortex it originates. Most, about 80% of them originate in the occipital cortex, so visual. And we don't know for sure because it's not really been studied well, but the more general idea is that the astrocytes within the brain that are obviously there for buffering, they're different in different regions of your brain. It's not one type across the whole brain. That even within the layers of your cortex, it's different types of astrocytes in different layers. And the visual cortex has a different density and distribution of astrocytes. So it's felt that maybe it's a reduction in astrocytes within that cortex that gives you that susceptibility there. But it's really hard to drive a CSD, a cortical spreading depression, which is the underlying phenomenon of aura, in like an animal brain. Even if you're doing that kind of work, you have to really drive the system, and you have to go in with some like chemogenetics and perturb the system, make it more hyper excitable. So more glutamatergic transmission, for example, or less GABA, and then you can start to tip. Because of course that's different from a human brain, where you've got the genetics of being a migraine sufferer, where your brain's already a little bit more hyper excitable, and therefore you can tip into these features. And of course we rely so much on visual stimulus for everything we do, that it's probably a mix of both that anatomy, but also how reliant we are as humans on visual input.
I have a question about the use of animal studies. Obviously, the big problem when you do like a lot of research is like how can, like for example, depression, like how can you tell if a mouse is depressed?
Yeah.
I was just like wondering, like seeing the symptoms of migraines in a mouse, like the aura, and like how do you know if it's having visual disturbances?
Yeah, so great question. The answer is, 100% you cannot model migraine in a mouse. But as I said, say thinking about the allodynia, not me because I don't have hair, but if you ask someone with migraine, they'll tell you that even brushing their hair feels sore, or touching. If you've ever had sunburn, for example, you'll have felt allodynia, that bit where the gentle touch is now a little bit noxious. And that's one feature that migraine patients report. And if you give a human with migraine a compound called nitroglycerin, about 90% of those will have a full-blown migraine attack about 2 to 3 hours after. If you give a mouse or a rat nitroglycerin, which by the way is the active ingredient of dynamite, but we don't use it for that purpose. It's also nitric oxide donor, so it's via nitric oxide thing. If you give that to a mouse, about 1.5 to 2 hours after you give it, they'll become hypersensitive to touch, hypersensitive to light. So they'll avoid touch, they'll avoid light. So as a marker of photophobia, for example. So we have behavioural apparatus, for example, like dark boxes, where we expose the animals and they can move between the light and dark as much as they wish. And we look at that in control, and then we give them these stimulating agents, or we stress them, for example, because stress is important for migraine. And they'll spend more time in the dark, out the light, just like a migraine patient would withdraw from the bright room and go sit in a dark room during an attack. So it's about using these translatable features that give us some indication of the biology. And of course, it's helped then if those features are inhibited by migraine therapies. So if we can show that the animal becomes photophobic, and then if you give, say, a triptan, and it now targets that photophobia, that gives us a reliable sense that this is a reasonable readout, but it's never 100%.
Yeah.
So it's best case scenario. And really a lot of what we do is not about migraine per se, it's about understanding how these brain circuits work in health, because only in understanding how they work in health can you actually understand how they work in disease. And I think often we've jumped forward to disease without really understanding how these things work in health. So the disease is the next level.
How did you find out that compound that's in dynamite causes like migraines afterwards?
People working in the explosive industry reporting. So if you're a healthy control and don't get migraine, I've had it because I don't get migraine, you'll get, for about 10-15 minutes, you'll get this throbbing pulsatility. And I don't consider it painful, but some people do say it's painful. It's like a headache. And then that goes away. That's the acute vasodilation, all the activation. But 2, 3, 4, 5 hours after, that's when the migraine person gets a full-blown attack, because it puts in sequence, it increases like cyclic AMP and all these second messengers in your neurons, makes them more excitable, and then triggers you into a full-blown migraine later on. So it was, again, just a kind of a side effect of people having migraine. It's not just nitroglycerin. We can do it with calcitonin gene-related peptide, for example, but only about 60% of patients will trigger with that. So different molecules we use as triggering agents. So we have both human models where we trigger in people, put them in the scanner, and then we take those molecules and put them in animals where we can then look in the brain to understand, for example, like in the scanner we see a bit of yellow light on the hypothalamus. What does that actually mean? does that mean 10 cells were activated or 1,000 cells were activated? And that's what the animal work allows us to do. It allows us to say, well, if these cells are orexin cells, what's happening to them and how might that be? How could we develop new treatments to target those cells to fix the condition? So it's always about, and I should say as well that the animal work's never done in isolation. It's always done in collaboration with human work, reverse translating phenotypes, cellular work, and like human iPSCs and things. So we only go to the animal for those really important studies that need to be done in the living organism. Everything else is done by modelling or cells or human induced pluripotent stem cells, for example.
For the stem cells, like, would you use them to like make organoids or just like test individual compounds?
We are not doing that at the moment, but we are starting to work with people who are making 3D organoids because, of course, and they make these, I don't know if you've seen them, they can make them nowadays on a balloon.
Oh.
So that they can have them pulsing to mimic like the pulsatility you get of the vasculature and things. So again, starting to think about those 3D organoids now. People do them either as co-culture organoids or just co-cultures, but also now on a chip where you can record electrical activity, or on a balloon, for example, where they can pulse it and you get that pulsatility that you would see in the brain naturally. So yeah, there's really advanced stuff. We collaborate with people from Leiden who are doing that work in the Netherlands.
That's really cool.
I've heard about organs on a chip, not on the balloon.
Yeah, the balloon one's just to give so that you can introduce that kind of rhythmic vasculature and pulse that you would get in a natural environment. So trying to mimic as closely as possible. Like anything, you can't say migraine in a mouse, you also can't say migraine in a dish or migraine in an organoid, but you can look at how the different cell components talk to each other. And again, you can use a lot of these imaging techniques on those cultures as well. The principle's the same, it's still calcium dye, fluorescent activation.
So with your work and everything you do in the lab, do you think anything, well, obviously at some point, all the research and the new information you're learning, and there is so much that you put in that presentation that genuinely is like, it completely changes what we know about pain and migraine in the brain. Do you think there's any work that could potentially happen in your lab or a future lab that you might run to do with, okay, well, we understand these new things about migraine, how can we use this to sort of tackle it?
Yeah, 100%. I mean, so I work with Peter Goldsby here at King's College London. He's a clinical, but also PhD. He was actually my PhD supervisor at UCL back in 2000. He won the Brain Prize 2 years ago, the Lundbeck Brain Prize, for developing the new calcitonin gene-related peptide therapies for migraine that are used routinely now for routinely, yes, and certainly there's expensive because some of them are biologics, some of them are small molecule drugs, but they're available now in 87 countries treating migraine around the world in people. And, you know, that's what we all aspire to do. What I aspire to do is, you know, try and come up with a way that my important thing is, rather than treating the pain or treating the attack, can we actually find disease modifying treatments? Wherefore, instead of having to worry about treating the attack, we can either reduce significantly the number of attacks or stop them completely. And that would be the ultimate goal, of course, to try and work. And then we've been doing some work recently where we've been looking not just at migraine, but also in like autism and ADHD, because of course these are conditions that also have sensory hypersensitivities to them. So can we learn from each other in terms of how we might cross-fertilize in those areas, and what we know from migraine might help in autism and ADHD and vice versa, because they're much better at phenotyping sensory abnormalities in people, and the ADHD field, we're much better at developing treatments, and we're trying to work together to help each other really. So we're trying to get funding for that just now. You had your hand up.
So would you say in your lab or in your research as a whole, do you implement much AI tools, or do you think there's a space for it?
I think there's a huge space for it, but I'm a bit of a dinosaur. And like, you know, I think you've got to be good at something. You can do a lot of things, but you've got to be good at something. You can't be good at everything. And a really important thing about being a PI or doing something is knowing your limitations. And I think a bit like we were speaking earlier with social media, I know my limitations. I've not gone there. You know, I think my lab has an X account, but the students deal with that and stuff. You know, it's just like that. You know, I don't know enough about AI myself to commit to it, but I think both clinically and pre-clinically it's a huge tool. I mean, we use some AI-based tools for analysis, you know, because it's very good for pipeline analysis of EEG, for example, because if you sit and do it manually, for every hour of recording, it takes you an hour to score that. So if you record somebody overnight, you're going to spend that same number of hours, so that it can make things much quicker and better. And then when we look at migraine, one of the big questions in the field now is, we've got these new therapies and they work great in some people, but they don't work very well in others, and we've not been able to predict that. I think that's where AI can come in. If we get enough data sets and we can share that around the world between people understanding the phenotypes of people that respond to drug X versus people that respond to drug Y, and use AI tools to unbiasedly search for patterns that we as people naturally miss. I think that's where it's huge here. And like, it's a big thing. King's have been putting a lot of money recently into recruiting people to drive AI components now in research. So they've been recruiting at least 6 or 7 new AI-driven fellowships at King's. So it's a really cool idea. It's a really cool area. We collaborate with a chap in Norway who is huge in it. They run a program called Norhead and headache, and I rely on them because they know much more about it than me.
And do you do a lot of the work with like EEG and like the very neurotechy side, talking about the like open source code for your probes and bits and pieces? Do you do a lot of that yourself? Is that a skill that you've acquired recently, or do you work with a lot of other people who have more of a technical background?
I've always been the technical person, so building things was always my background. I grew up in farms, so in rural farms, so you kind of had to build it, make it, or fix it. So that was always my background. So I had 5 sisters, and I used to spend my life taking apart. You guys won't have had like tape decks or Walkmen. So like, I remember in high school I had to, the one where you put the colour disc and you put all the colours of the rainbow, and when you spin it, it goes white. So we did that as a science experiment. So I went home and got my sister's like Walkman that played tapes, which has a little spinny bit that spins the tape and thread. So I took that and then dismantled it and made that into a bit that I could put the disc on to spin the rainbow. And it was great. Went into school and they were all very happy to see the experiment. My sister wasn't so happy when I broke her Walkman. But so I was always into that technical building side of things, and I think if I hadn't been in science or like neuroscience, probably engineering is something I would have liked. Where I've not been as good as the coding side of it. I can change code. So like, we all share code, and I can kind of take a piece of code and share it and say like, no, I want this code to do that for me. But when it comes to writing it, I'm not so experienced. So everyone on my team that I recruit, all the new students, they all get sent on like a coding program. It runs for about 18 months in the background while they're doing their research. It's run through, the one I use is run through UCL. It's called Seismic, and it's a practical one. They do projects and develop. But again, it's really no point in them learning it until they're doing it, because then they're getting the practical experience, and it's really about future proofing for them. For me, I don't need to be able to code, because they don't let me in the lab too much now unless something's broken and I get in to fix it. But they're the ones doing that work, so it's about facilitating it for them now as a PI, not so much me learning those techniques.
Okay, any more questions? Okay, brilliant. Well,
Yeah.
we
Thank you, everyone. And
let you get back to get you to your son's football match. Thank you so much. Can we all just give him a round of applause?
Thank you.
And hopefully, I'll get in touch with you about some of us coming to see the lab. It sounds
yeah,
really
we're just building a brand new fibre photometry system at the moment.
Field trip for 3 of us. We'll do a giveaway. You
Yeah,
can have
we're
a free
just-
spot to Phil Holland's lab.
We're just doing the Gen 2 fibre photometry system, so we're hoping to have that built before Christmas and be doing recordings with that mid to late January. So once that's set up, people will be welcome to come and see it, for example, just to understand.
Brilliant. Thank you so much. And thank you if you joined online.